Greetings and welcome back as we continue our exciting journey through the world of nucleic acids. When we left off last time, we were talking about DNA replication, deoxyribonucleic acid, and now we're going to move on to DNA's little helper called RNA. Now hopefully you guys remember that DNA within its nitrogen bases, the A, T, C, and G, the order of those letters in DNA has the code for making proteins. Now, proteins are super important because they control everything that your cells in your body do. So, since you're made of cells and proteins control your cells, proteins make you who you are. They give you all of your traits, your characteristics. They make you look how you look and act, to a certain degree, act how you act. That's all because of proteins. Now, unfortunately, proteins are made inside of organelles within the cell called ribosomes. Proteins are made within the ribosomes. Now, you're probably saying, well, what's the big deal? Okay, DNA has the code for making proteins, and proteins are made inside of the ribosomes. So what do we need RNA for? Well, DNA has one little tiny problem. DNA is kind of like in jail. It is stuck inside of an organelle called the nucleus of the cell. So since DNA is stuck in the nucleus and proteins are made inside ribosomes, which are outside the nucleus, DNA can't directly make proteins itself. But what can it do? It can hand off its code to its buddy RNA. RNA can travel inside the nucleus, and it can leave the nucleus to the ribosomes. So RNA carries DNA's code to the ribosomes, which then can be used to make proteins. All right, so RNA stands for not deoxyribonucleic acid, but ribonucleic acid. And it's called that because its sugar is not deoxyribose, like DNA had, but its sugar is called ribose. It's basically the same thing as deoxyribose, it just has one more oxygen in it. All right, so now, function of RNA, like we said in the previous slide. RNA copies the genetic information from DNA and takes it to the ribosomes so you can synthesize or make proteins. Now that's one of the jobs of RNA. That's its main job. Now look, there's 3 types of RNA. 2 of them are going to be super important. So if you're like, I can't remember 3, I can only remember 2. All right. Well, definitely remember this one, mRNA, which stands for messenger RNA, and remember tRNA for sure, and that stands for transfer RNA. Now, you should be able to remember 3 things, and this is RRNA, and that stands for ribosomal RNA. Now, if you can see down here, this little shape right here should definitely look familiar. You should be like, hey, I've seen that before. Even if you don't know what it is, it should look familiar. Well, that is an RNA nucleotide. Same thing as DNA nucleotide, just has pretty much one difference. It's still got the phosphate, just like DNA. It's still got a little thing that looks like a house, but instead of a D inside there, it's an R because it's ribose sugar and not deoxyribose sugar. And it's got one of 4 nitrogen bases, just like DNA. But there will be a slight difference when it comes to the bases in RNA. Just a slight difference, so don't get all worried. All right, so the first type of RNA we're going to talk about is messenger, or mRNA. Messenger RNA and its function, we're going to talk about this next week, but transcription, that is the process of RNA copying the information in DNA in the nucleus, and then it takes it to the ribosomes to make proteins. So messenger RNA is the one that copies the code in DNA within the nucleus and takes it to the ribosomes. And over here you can see a little tiny little mRNA molecule. Here's a little nucleotide, RNA nucleotide there, with its base guanine or G, little house with ribose sugar, and its phosphate. Now, if you can see here, one difference between DNA and RNA, RNA is single-sided, so it's not a double helix, it doesn't have two sides like DNA does. It's single-sided, so remember that. You have to remember the differences between DNA and RNA. Another difference between DNA and RNA is this base right here. Whoa, U, where'd you come from? So with RNA, it's not A, T, C, and G. It's A, U, C, and G. All right, so right here, you have a little longer messenger or mRNA molecule. Messenger RNA. Now, this guy gets made inside the nucleus, obviously, because it's copying the code in DNA to make this mRNA molecule. So, what letters would the DNA have to make this exact sequence in the mRNA? Well, the DNA would have this sequence of letters. T would make the A in mRNA. Then it would be A gives you the U. C goes with G. A goes with U. A goes with U. G goes with C. T goes with A. T goes with A. T again goes with A. G goes with C. A goes with U. And G goes with C. So, this sequence of bases in DNA would make this exact sequence in a messenger RNA molecule. And that's basically how messenger RNA is made. DNA unzips, just like during DNA replication, but instead of making another DNA molecule, you make a messenger RNA. Now, what base is missing from mRNA that is found in DNA? Well, remember, there's no more T, so T is no more. But there is a U, which stands for uracil. Uracil. And what base does uracil match with in DNA? Well, there's no more A goes with T, but it's going to be A now bonds with U, and C will still bond with G. So, C and G stays the same. There's just no more T in RNA. We replace it with U or uracil. All right, so inside this little red little oval here, that's the nucleus. This right here is a strand of DNA. It has the code for making proteins. So that little strand of what? 6, 9 bases or letters in a row has the code for making a very, very small little protein. Now, but remember, DNA is stuck inside the nucleus. It cannot get out. But it's got to get the code over here to this ribosome, because that is where proteins are made. Proteins are not made within the nucleus. It's got to get its code to the ribosome so it can make its protein. So with that code, what series of letters or bases is going to be in our mRNA molecule? Well, that's easy. It's just the base pairing rules. But remember, we're going to get rid of T, and we're going to replace it with U in RNA. So, T will go with A. A will not go with T now. A will go with U. C still goes with G. G still goes with C. T still goes with A, because we still have A in RNA. C goes with G. A doesn't go with T. A goes with U. C goes with G. And finally, G goes with C. Boom. We made our messenger RNA, and now that mRNA can leave the nucleus, take DNA's code to Mr. Ribosome, where we can construct a protein molecule. All right, so the other two types of RNA. This is the middle one, the one I said if you cannot remember 3 things, if you can only remember 2 things. This is the one you can forget if you can't remember 3 things. But RRNA stands for ribosomal RNA. And basically, all you need to know is that ribosomes. Remember, that's where proteins are synthesized or proteins are made. They're made within ribosomes. And ribosomes are made mostly of ribosomal or rRNA. That's all you need to know. Now, unfortunately, you cannot forget tRNA because tRNA is pretty important. Now, if you remember, messenger RNA carries the code from DNA from the nucleus to the ribosomes. But its buddy, tRNA, carries the ingredients or the building blocks of proteins to the ribosome. And those building blocks are called amino acids. Hopefully you guys remember that. Remember, proteins are made from the monomers of the building blocks, amino acids. So, mRNA carries the code. mRNA carries the code to the ribosome. tRNA carries the little building blocks to the ribosome. And when you put both of those guys together, the code plus the things that make proteins together at the ribosome, what do you get? You get proteins. Alright, so transfer or tRNA. First of all, if we remember from the last slide, their job is to carry amino acids. So we see a tRNA right there carrying the amino acid called leucine, specific type of amino acid. And this other tRNA is over here carrying a different type of amino acid called phenylalanine. So that's the amino acid right there on top. So tRNAs carry the building blocks of proteins to the ribosomes. Now, if you look at these words in these sentences, words in sentences can be made of a lot of different numbers of letters. Like acid is made of 4 letters, at is 2, the is 3. Well, one thing you need to know, RNA, all their words that they talk with are always 3 letters. Never 2, never 4, never 7. So, you can see these 3 letters on the bottom of these tRNAs. This GAC right there, GAC, and this AAG right there, AG. Those are called anticodons. Anticodons. 3 bases on the bottom of a tRNA molecule makes an anticodon. Why is that important? Well, remember that mRNA molecule we made a few slides back that was 9 letters or 9 bases long? Well, these anticodons on the bottom of the tRNA, they're going to match up with 3 letters on an mRNA molecule. Now, those 3 letters on an mRNA molecule are not called anticodons, they're called codons. So 3 letters on an mRNA molecule makes a codon. Alright, so what would the codon be that would match up for this tRNA's anticodon? Well, obviously, A goes with U, A goes with U, G goes with C. Boom. That tRNA would drop off phenylalanine, the amino acid phenylalanine. Alright, what would be the next 3 letters in the mRNA? Well, G goes with C, A goes with U, C goes with G. Boom. That tRNA would drop off the amino acid leucine, and so on and so on. So, just remember, tRNA and mRNA, they always talk in 3-letter words. The words in tRNA are called anticodons. The words, or 3 bases in a row, in mRNA are called codons. Alright, so this is a pretty important slide because this will eventually be your guys' favorite type of question on any exam. And we call those either essay or short answer questions. Now, the differences between DNA and RNA. What are their differences? Alright, like we said earlier, DNA is double-sided. RNA is single-sided or only has one side. DNA has deoxyribose as its sugar. That's why it's called deoxyribonucleic acid, or DNA, where RNA has ribose as its sugar. In DNA, we have thymine that matches with adenine. RNA, we have uracil. Uracil, or U, that matches with adenine, or A. And last but not least, DNA, if we remember, poor little DNA is trapped in the nucleus where RNA can move around the cell. So those are your four main differences between DNA and RNA. Alright, so our last slide. I'm sure you're very sad to hear that. You're like, oh, come on, we want to learn more. This isn't enough. Well, don't worry. There's always next week. Alright, so the genetic code in action. Genetics, your genes, what makes you you, what gives you all your traits. Well, it's your DNA, which has the code for making proteins. Remember, proteins are made from their building blocks, amino acids. Now, different proteins, we're all different because we make different proteins. Well, what makes proteins different? Well, it's the sequence or the order that the amino acids are put in place. These proteins are thousands of amino acids long. Well, it's the sequence or order that those amino acids are bonded together, which makes the proteins different. And like we said earlier, since proteins are basically, the main molecule of shape and structure, and they control all the chemical reactions within cells, proteins basically control who you are. But who controls the proteins? Well, it's DNA, the code in DNA, which controls everything. Because DNA controls which proteins you're going to make, and that gives you what's called, you won't need to know this yet, gives you what's called your phenotype, and that's just basically what traits you have. So if you have brown hair, that would be your phenotype for hair color. How did you get that? From your DNA. How did your DNA give you your brown hair? Well, DNA made a specific protein, which gave you your phenotype or your brown hair. But for right now, don't worry about genotype or phenotype. We're going to go over that later. Just know that DNA controls the proteins, which thus controls what type of traits you have. Thank you very much, and I'll catch you next time.